it's important to recognize that migraine is more than one disorder. So we know that from person to person with migraine, there are differences in symptom profiles. One person has nausea, another has photophobia and phonophobia. We know the differences in attack frequency. We know the differences in comorbidity profiles. We know the differences in prognosis. And we know the differences in genetics. So for rare forms of migraine, like familial hemiplegic migraine, we know that there are 3 genes that account for the majority of familial hemiplegic migraine. But for ordinary forms of migraine, more than 40 genetic variants have been identified, and those 40 genetic variants account for a small proportion of all migraine. We also know that there are huge individual differences in treatment response. For one person, topiramate may be a godsend. For another person, it may be unhelpful and produce a lot of side effects. One acute treatment works for one person and not for another. So all this means that migraine is a highly heterogeneous disorder, and one of the hallmarks of clinical trials is that we, for the most part, ignore that heterogeneity. We lump everybody together in a big pot, and then we say, well, why is it with the best of available therapy only 50% of people respond? So I think one of the major steps forward we need to take is figuring out ways of identifying more homogeneous groups of people with migraine based on symptom profile, based on treatment response, based on prognosis, and develop a clinical strategy for identifying those individuals so we can do a better job of getting treatment right the first time and avoid the process of trial and error therapy, which can be frustrating both for doctors and patients. So one area we need in clinical trials is learning enough about patient groups to identify homogeneous groups that do and don't respond to particular treatments, who do and don't carry particular genes, and who may differ in their longitudinal trajectory over the course of their lifespan.
We have a lot of different drugs for the treatment of migraine, but we really don't know which would be the first and the best to use. So we're looking for markers, and what we don't have are many comparative trials of the older drugs versus the new drugs. And lastly, combination therapy. Which drugs work better together and which drugs don't? So for example, we did a study a while ago adding topiramate to a beta blocker, and 2 of them didn't work any better than topiramate alone. But in the fremenzimab studies, we allowed the concomitant use of topiramate, and those patients continue to improve. And they improved just as well, people off of it. What we don't know is what the additional benefit was, but we know they did just as well. So we need biomarkers, we need new targets, and we need combination treatment. New targets, people are now looking at the marijuana and its derivatives for the treatment of migraine. That's an ongoing field. The major problem we have, it's still considered illegal by the federal government, so it's very difficult to do clinical trials on a drug that's considered that, even though more and more states are legalizing it. There's new targets like PACAP and the delta opioid receptor. A lot of those are coming in development.
At the moment, the most promising agents for change are going to be in the preventive space are going to be these CGRP monoclonal antibodies. We have 1 available now. There are another 3 coming along, the 3 peptide ones, eftanizumab, fremonizumab and galcanezumab, to add into arenumab. So we're going to have those choices. I think that the development of the small molecule CGRP receptor antagonists, ubrogepant, rimegepant, hugely useful. Lasmidotan, serotonin 1F receptor agonist, hugely useful in acute therapy, getting away from the vascular side of things. Coming along after that, there's work that's exploring pituitary adenylate cyclase activating peptide, PACAP, at the so-called PAC1 receptor that's in clinical trial. And there's an evidential, there's some clinical work and laboratory work to make one think about metabotropic glutamate receptors, to make one think about orexinergic mechanisms, acid-sensing ion channels, nitroergic mechanisms, the TRPM8 channel. What's really exciting about migraine at the moment is putting all these medicines, of course, neuromodulation approaches, non-invasive vagal nerve stimulation, transcranial magnetic stimulation. There's an incredible collection now of mechanism-based ways of thinking about what's going to happen both in acute and preventive treatment. I don't know what everything we're going to have at our hands, at our fingertips in say 5 to 7 years, but I do know there'll be more. I do know they'll be effective. I know they'll be well tolerated, and I know a lot of patients are going to be much, much better, and physicians are going to be much more satisfied with their practice, particularly in headache medicine. I think through clinical experience and some well-designed trials, we're going to need to learn how to use these new therapies that we have. We need to establish starting rules and stopping rules. We need to try and take the next step in many ways. I think this is true of acute and preventive therapy, in personalising the treatment. I know if I take 100 people with migraine and I give them a CGRP pathway monoclonal antibody, whether it be a ligand or receptor, about half of them will respond. I know about a third of them will have a 75% response. I don't know who they are. So one of the most important things we need to do is to move into an era where we can personalise the treatment we offer. Why should I give you Rimagepant or you Ubrogepant or you Lasmidatan? Why would I give Erenumab here and Fremenis? Why would I do that? At the moment, we diagnose migraine, chronic migraine or episodic migraine, and we use these treatments. I would like to see a world where we understood better who should get what, so we can get it right first time for patients, so they don't spend so much time moving around with failed medicines.
I work in two areas. I work in migraine and I work in Alzheimer's disease. And so it's been fascinating to go to migraine meetings and to be in a session where we're presenting the 5th positive randomized trial in a single session, and people are sort of bored. And then I go to Alzheimer meetings where nothing has worked for a decade, and even a hint of a subgroup analysis with a positive result is a source of tremendous excitement. So I appreciate that these are really amazing times in migraine, that there are multiple devices, multiple drugs as acute treatments and as preventive treatments that are emerging. You know, I recognize there are 1 billion people globally who have migraine, that migraine is the world's 2nd leading cause of health-related disability after low back pain. And I'm very excited to be part of the process of bringing new treatments into the clinical sphere, which I believe will materially improve the lives of an uncountable number of people all over the globe.